online people, we just watched those first 3 YouTube videos that are in the PowerPoints, and we're going to then talk about all of those things. We've already got, because of last class, we already know about getting signal to muscle, and now we're going to talk about exciting muscle and how muscle responds. Alright, so where we were, right, we'd had acetylcholine in the synaptic cleft, right? Great. So what's happening here is the acetylcholine from motor neuron binds the acetylcholine receptors on the sarcolemma. Remind me what the sarcolemma is? Yeah, it's the thing that surrounds the muscle fiber, right? It's the plasma membrane, but a muscle, and so it's fancy. Okay? This is going to open an ion channel, or lots of ion channels. In other classes, have you seen ion channels before? Right? Okay. They're voltage gated or ion gated or whatever. These ones are ligand gated. Okay? Meaning once this acetylcholine sticks onto it, it opens it up. The acetylcholine itself isn't entering the muscle cell. Okay? It's like, if somebody is delivering packages, they can come up, ring your doorbell, they stay outside, but them being there sends a signal inside, and then you open the door. Okay? But the acetylcholine itself isn't entering the muscle cell. It's a really common misconception. These ion channels are going to open, and some sodium that's got a positive charge on it, right? A sodium ion is going to move into the muscle fiber. Some potassium is going to go out, but not as much as sodium's coming in. Okay? So you're going to have some positives going out, some positives coming in, but more positives entering than leaving. So now somebody gets more positive. All right? So the interior sarcolemma there is held at a negative potential. It's got a negative charge to it. It gets less negative. All right? It's called depolarization because it used to be far away. It used to be zero here. It used to be really, really polarized, but brings it closer to less polarized. This is called the end plate potential. It's a voltage potential. And that's the picture here, right? Lots has happened. Sorry, online people. Let's make it so you can see this. Sorry. Anyways, I hope you're following along with your slides. Okay. Calcium. We've dumped acetylcholine out. It's come here, hit these receptors. Sodium's gone in, and now this is depolarized. And that depolarization is going to spread. What happens is you have these ion channels that were ligand gated. Cool. You need acetylcholine to open them. So if there's a little bit of acetylcholine, it might not open it, but enough acetylcholine definitely will open. But on the muscle fiber itself, there are a whole bunch of voltage-gated ion channels. Kind of like the voltage-gated ion channels we see here for calcium, right? The action potential comes, there's a voltage change that opens this and calcium can come in. Along here, along the muscle cell itself, we've got these voltage-gated channels. So what's going to happen with that is if enough sodium has come in because of acetylcholine binding, and the whole sarcolemma thing now is depolarized enough, you'll open the voltage-gated channels. Is everyone with me here? Okay. Those are what's going to actually make lots of things happen. So sodium is going to pour in through those. And once those voltage-gated sodium channels open, there's no turning back. All right, like at that point, you're going to have this action potential along the muscle. And it spreads really, really quickly. This action potential is going to spread from one voltage-gated sodium channel, right? It opens, it lets sodium come in, and that makes the next portion less depolarized, right? Sorry, less polarized. Depolarizes it. So that part opens, and it's going to spread both directions really fast. We're okay with this? So, we've had this end plate potential. This got depolarized. As that got depolarized, can you see this, people? Sorry. As it gets depolarized, we're going to open these voltage-gated sodium channels. And on this picture here, you'll see a couple things. See those little sticky out bits? Right? This part of the protein responds to the voltage on the membrane. So if the inside's negative, which is normal, right? These are held down, like held down and it gates the channel. When this is depolarized, when this turns positive for a second, it pushes those up, right? So it's voltage gated, right? If this is very, very negative, it's closed. If it gets positive, it's open. You'll also note that there's this little ball and chain thing going on here. It legitimately looks like a ball and chain. That's going to come important later. The potassium channels, hi people. Potassium channels here are also voltage gated, but they're slow. They're trying to respond to the voltage change, but it takes them a couple milliseconds to get going. Yeah? So far so good? Okay. So, back here with this little ball and chain situation, this is going to set up the repolarization of this whole muscle cell. So, those voltage-gated channels close, actually they inactivate. That ball and chain is kind of flopping around, and after a certain amount of time, just because of random motion, it plugs the hole back up. Okay? So the voltage part of the gate is still open, but this ball clogs the hole. Does that make sense? I feel like I maybe lost some people. So long as, okay, there we go. I think I lost people, but stop me if I'm just repeating myself too many times. I should draw, but it's really hard to, yeah, I'll try drawing. And this one. Okay, let's see, I hope. Sorry, I realize I fiddle with this bunch, but it's... Okay. We've got sarcolemma. Okay, got it there? Great. We have in there, we've got voltage-gated sodium. Okay? Voltage-gated sodium chain. It's got hole sodium can go through. Normally it's gated. When this is negative, there's, I don't know exactly the best way to draw this. Yeah, when this is negative, there are gates that are closed, okay? As soon as this goes positive, that pushes this out of the way, and this whole thing's open, right? And this hole, which was been there the whole time, can let sodium through. Great. Okay? Once this goes back to being negative, this is going to close again. But as long as this is positive, they're open. Yeah? There's the protein for all the world that looks like a ball and chain. It's got a really, really thin little polypeptide string, and then this glob of protein right here. What it does, it's just kind of flopping around, and when this whole thing's closed, it just like hangs out. But when it's open, it flops around because it does, and it'll plug this hole. Right? All right, plug that hole. That hole now is closed. It's open because of the voltage, right? But sodium can't get through because there's this blob in the way. Okay? This blob is going to probably depend, but for our intents and purposes, this blob is going to stay there until this repolarizes. So this is going to go negative, right? These gates are going to go back and close up. As those gates close up, it's going to kick the ball out. So far, so good? People who've seen neurons, you've seen a similar thing happen. Okay. We're good? Great. So as this is all going down, the potassium channels have finally like woken up. Oh gosh, you get going. And the potassium channels are going to be pouring potassium out of the cell. Right? So as potassium leaves with this positive charge, it's going to repolarize. Right? Yes? No? I can't tell people's eyes. We've had it in like faculty meetings, they're like, okay, so you get less visual information from your students when they wear masks, so like watch their eyes, which I feel creepy doing. I'm like, hi. But let me know how we're doing. So that's where we're at. This potassium leaving is going to pull it back to the initial resting membrane voltage. And for a little while, the muscle cell can't do anything. Because you've got the sodium channels clogged up and the potassium channels are open and moving stuff out, even if you throw more, even if you zap the muscle again, it couldn't do anything because it's already busy. You can't have this happen again until you've, until you basically cleared the system. So far so good. The refractory period's pretty short. That's okay. And then you might have noticed there's been sodium potassium that's moved now across the sarcolemma, right? There are ion pumps that just work round the clock. And so as they just keep chugging along, they're going to be shoveling sodium out and bringing potassium back in. They just chug away constantly. So these pumps are pumping sodium out, pumping sodium out, pumping sodium out, pulling potassium in, potassium in, potassium in. Stimulation happens, sodium rushes in, potassium rushes out. The pumps reset that. Yeah? We're good? This, amongst other reasons, is why, like, if you're doing all sorts of exercise or working hard with your muscles, you need to make sure that you have some sort of ions in your system, right? Like, you're going to lose a bunch of salts through sweat. You need to make sure that, you have some sodium happening here. And here we're back to everything's back to normal. We're good? Okay. So this, if we're watching the membrane potential, we had it just a resting potential. We had just a resting potential here. Sodium channels open. The voltage gated sodium channels open, right? the cell gets less and less negative because sodium's coming in. The sodium channels are going to close up. The potassium channels open. Potassium channels opening are going to repolarize the cells. Potassium's still positive, right? But if this positive ion's moving, how come the cell's getting more negative inside? Because the positive's leaving. Okay, is everyone fine with that? Yes? Thank you. Yes, this is important. Okay, so positive leaves, potassium channels close, and we're basically back to normal. Yeah? Okay. Great. You know, this PowerPoint here that's supposed to be downloading, I should always use my office. I just forget. It says it's going to take only 50 more minutes. Yeah. Anyways, the next bit, now that we've had this action potential go along the muscle cell, things happen. I was thinking about having people memorize these things and like act them out, but we'll save that for later. Several different parts here. This video is the second one we watched, so if you're studying and you want to go over it, that's that one there. Okay, we had the sarcolemma, which was basically the plasma membrane. We now have the sarcoplasmic reticulum. It's basically the ER, but in muscle cells, okay? It's going to surround each of those myofibrils, right? Those long little organelles. They're going to run, generally, we're in skeletal muscle here. They're running long ways, okay? And, yeah, so many terms. The SR is mostly going to worry about calcium inside muscle fiber itself, okay? So calcium, again, at the neuron was important for acetylcholine, but we don't care about that now. We're inside the muscle fiber. The SR is going to hold and release calcium. It's hugely important for this. The ER handles calcium just in regular cells too, but for muscle cells, we really, really need this to work in a very particular way. So yeah, that's the SR. There's a part called T-tubules. I'll show you a picture in a sec, but just I want to talk about it. The sarcolemma, the plasma membrane, goes deep into the cell's interior. So that's going to increase the surface area dramatically. All right? It's kind of like, like a ball has surface area, right? Has some area. A pine cone has a lot more surface area, right? Because it goes in and comes out, right? T-tubules are going to be that. The sarcolemma is folded in deep, okay? Huge surface area. And this is going to be really important because that electrical potential is going to be running fast, that action potential is going to be running fast along the, along that membrane, right? And so if you have that membrane coming in deep, that signal can get deep into the muscle fiber. Fair enough? And then there's a portion called the triad. Just for the record, the triad's also like a Chinese gang, but this is a different thing. Chinese, Japanese. I think it was Chinese gang. Anyways, it's the T-tubule. It's this. Oh my goodness, words. It's this situation right here, where you have the T-tubule. You have the terminal cisternae. You've got this whole chunk right together. It's called triad because there's the three parts to it. And we see how yeah, we'll talk about it. We'll see how right here we're inside one muscle cell. Okay, so again, the sarcolemma is the plasma membrane of the whole cell. All right, these little myofibrils, they're organelles inside that one cell. Okay, yeah. These are all in the same cell. And you see here this T tubule right here. It's shown like this. What it is, it's the outside. It's the sarcolemma that's dug in deep. Yeah? Lost anybody? We're good. It's dug in deep, so you can't see some of the sarcolemma because it's right here that goes in. And all this SR here, is going to be doing a really important job for holding on to calcium and worrying about it. You'll see there's a ton of mitochondria floating around as well. That's important. The big news that happens here is this T-tubule has proteins that notice voltage, right? Everything's noticing voltage here right now. This is why amongst other things, tasers work, right? Like, muscles respond to electricity in dramatic ways. If there's an electrical current, right, because of this action potential, these tubule proteins notice that. And they're going to change shape, all right? And they've got here the, oh gosh, I need to focus. Sorry, I was up all night. Okay. These integral proteins that are in the SR, those are going to control calcium, right? They're going to be controlling the calcium channels. And so the take-home message there is an electrical impulse comes along, zoom, right? The T-tubule proteins are going to change shape. They're going to bend and move, right? So electricity made that move, right? Once that moves, that basically shoves or pokes those SR proteins to change shape, which are then going to dump calcium out, right? Couple steps here, but the end result is that electrical current moving along releases releases calcium out into the cytoplasm of the muscle fiber, right? So, zoom, some electrical signal happening along here on the sarcolemma, wet and nice and deep. Series of events, but the end result is this SR dumps calcium out. Cool? Great. So that whole point is just to get calcium hanging around here so actin and myosin can do stuff with it. Yeah? Okay. So we're now here. So we're now into the final portion of how this works. So we've gotten a signal from the neuron saying go. We've had the muscle fiber respond electrically, right? Like, oh, right, I got that signal. I'm gonna spread an action potential along myself. And now we're inside inside a muscle fiber, inside the muscle cell, and we're gonna actually have it contract. Okay? This is called the sliding filament model of contraction. Your lab talked about this a lot, right? It had like the person dragging on the big weight and yeah. Okay. Questions? Is this too fast? This stuff I think is cool. It's interesting. I just think that there's a lot of moving parts, and it's easy to sometimes switch parts, right? But looks like people are nodding and following along, so that's good. Briefly, but then there's lots more. Something called cross bridges are going to be made, and that's what's going to be contraction of the muscle. Okay, the muscle's going to contract. Your muscle will get tension in it, and once those cross bridges become inactive, the contraction stops. Things you don't want to watch it again. We've already seen it. Yeah, no, I showed it to you like 3 times now. This cross-bridge cycle, that film we watched at the very beginning, has 4 main things that are going to happen. It's going to form a cross bridge. There's going to be something called the power stroke, which sounds cool. Has anybody ever like rowed a boat? Yeah, okay. That motion, the like something's attached and then it pulls, that idea is the power stroke. The cross bridge is going to detach, and then the myosin head needs to reset, needs to be reactivated. These players over here, the sarcomere, calcium, tryptomyosin, they're the major players. You can go through all of them. This video, again, is the same one we saw if you want to study along with it. Okay. Got words? Do you want me to, is it helpful to draw stuff on the board? Or not? Are people happy with words? We're gonna go with words, and we'll draw if we need to, okay? If everything's just chilling, the muscle cell's just there because you're just not activating it, the intracellular calcium's low, which is good, so everything's just cool. Oh my goodness, yeah. Well, we'll do it here, We'll start right here. For your own personal study, there's a bunch of slides that talk through this, but I want to just show you pictures. Okay. Everything's cool, everything's relaxed right here. This is a sarcomere. We're now inside a myofibril, inside the muscle cell, right? There are, I believe the technical term is bajillions of these little guys, okay? All right? And they repeat and they repeat. This sarcomere is one sarcomere, the other one's another one, on and on and on, right? In your lab, when you were doing these, and you watched that muscle contract, did the muscle shortened, right? Did it get fatter? Or thicker at all? And at least one group was like, hey, it got chubbier. It does, right? Like, it doesn't disappear. But okay, so this is what looks like relaxed. This is what looks like contracted. And you'll note that, this relaxed one, this contracted one, there's going to be a whole series of sarcomeres all along, and they're all doing the similar thing, right? So this one's contracted, this one over here is contracted as well, and on and on and on, right? Question, and this isn't perfectly drawn scale, but this little actin piece here, has the length of a physical actin, this little blue string here that we're looking at, is that shorter than it was up there? That blue piece? No, it's not gotten shorter, okay? What about this myosin here? Is it as long as it was up there? The physical part? Yeah. So actin and myosin are both proteins, and they're not somehow like melting or shrinking. So how come this little sarcomere can get shorter? Because up there it's longer, and here it's shorter, but the proteins themselves haven't changed size. Yes, yes. So it's weird to like repeat everything, but it's hard for online folks. She said that they just move closer together, right? And that shortens the whole thing without, like, your fingers don't actually shorten when they just move closer together. They just there's less space between them. Okay, so everything's relaxed, everything's chilling. There's not a lot of calcium floating around here. It's all just relaxed. You've got myosin, you've got actin over here. When an action potential moves along, boom, calcium ions get dumped out from the SR, right? Calcium ion gets dumped out, which means calcium ions showing up the myofibrils is the final step for go for muscle. Okay, there've been a whole bunch of the series, but calcium ions showing up, that's what's going to make it actually go. Calcium ions clear a block. So in the video you watched, you saw how there are those binding spots that are clogged up, right? When calcium comes in, it clears those sites, all right? So now the myosin binding spots are open, they're available, all right? Myosin's the one that has that big head thing, the glob on it. The myosin binding sites and actin are then cleared. Myosin can then bind to it. It's great. It binds to it, like basically clicks in almost. Has anybody here ever used like a mouse trap or a mole trap? Where it like clicks? Does it scare you at all to set mouse traps? Like, you're totally a grown up, you can do it, but you're like, okay, you set it, and you're like, eh. No? Okay, maybe it's just me. It makes me nervous. Nobody else? Okay, apparently everybody else is like psychologically calm. There are other things that work like this too, like, I guess, jack-in-the-boxes, right? Like, it's wound, nothing's happening, but like, if you poke it right, it'll go. Yeah? The idea's there. You can think of the myosin heads kind of like being a set mousetrap. There's energy put into them, and they're in what's called the cocked position. So they're held back basically. They bind to myosin, and that makes it snap. Okay, it's like a mouse jumping on a mousetrap. It makes it snap. Yeah. When it snaps, it basically pulls along together. Yeah? We're okay with that? And yes, to then reset it, so it's caught, it snapped, it pulls along, and now it's done. To reset this myosin head back, you need ATP to basically ratchet it back. And so if you have ATP, it'll recock it. If the myosin binding spot's still open, it'll snap again and move. Right? Reset, snap, move. Reset, snap, move. Okay? That's why you probably all know I guess spoilers. Does anybody know what was in their unknown in lab? ATP, right? You needed to keep being able to reset that myosin head, right? To be able to have it go. Because if there's not ATP there, it's not going to happen. So that whole bit, now it's going to use words. Okay. You saw the picture, just make sure we're all on the same page. Tropomyosin is what's blocking the active sites on actin. Don't memorize it this way, but it's like tripping up myosin, right? It can't block, it can't bind there. So the myosin heads can't attach to actin even though they're ready to go. The T tubules change shape in response to voltage. The SR released calcium. Now, at when calcium's high, calcium binds to troponin. Troponin is going to change shape, and that's going to pull this tropomyosin away from the binding sites. It's going to clear them out. Myosin heads then attach. That attachment's the cross bridge. It's called cycling, just because once they attach, they do their thing. The sarcomere is going to shorten, the muscle is going to contract. Once you stop having action potentials running along, once the muscle stops being stimulated, the calcium's busily being pumped back into the SR. Right, kind of similar way. So like, typically there's low calcium concentrations floating around inside myofibril because calcium's being pumped back into the SR. Voltage changes will trigger the release, but it keeps getting put back. Myosin has high energy. Power stroke is when it pulls the actin towards the M line. ATP is what's going to both unattach and reset it, and that's going to be used for the next cross-bridge cycle. Yeah? Concerns? I think today is the most information-dense class we've had so far. But people are doing great. Yeah. Okay, that's the, like, microscopic physiology that we're looking at. I want to get you out like 15 minutes early to take your quiz. Are people okay to move on to another topic for about 10 minutes, 5 minutes? Just to kind of see it. We're going to now look at whole muscles a little bit more. We are not going to finish this because we're not. I'm trying really hard to keep this class kind of focused. I mean, myself focused. It's a constant struggle, but there's a lot to go over. So this is a whole new topic. We're on whole muscle contractions as a thing. And this finally downloaded, so that's good. Goodness. We are here. Wait for it. Okay. Whole muscle is going to be doing its thing. So, obviously, you can generate different amounts of force, right? So, it's a subtle point, but an action potential along a muscle fiber comes in like either on or off, like either the action potential is happening or it's not. All right? Either you're going to release calcium stores or you aren't. All right? But you might have noticed that your muscles, you can have different amounts of force, right? Right? It's not just either my muscles are on or they're off. Okay? So whole muscle contraction is set up by individual contractions, but there's a lot of things summing together in interesting ways. So it's going to be responding to both the frequencies and the intensities of the contractions. All right? So the tricky bit here is that you can use muscle cell as a term, right? Muscle cell, or you can use the whole muscle as something. Okay, so just there is difference. Here's a picture. We've got this whole big ginormous muscle. Great. Inside we have a whole bunch of these little muscle cells, and each muscle cell is going to be getting stimulated by a neuron. Great. A motor neuron can stimulate more than one muscle fiber, though, right? So this little red one is stimulating several different muscle fibers, but that's okay, right? For really, really fine control, like in your fingers, you're expecting that a motor neuron has a fairly limited range of connection. So this motor neuron talks to these two muscle fibers. Whereas someplace like your thigh, you're not worried about super fine movements. You're expecting a motor neuron to come in and innervate a whole bunch of muscle fibers. Be like, yeah, I'm going to tell them all the same thing anyways, just go for it. Yeah? This whole situation, the motor neuron plus all the muscle fibers, muscle cells it innervates, that's called a motor unit. All right? And you expect the motor unit to turn on and off together. So if there's a signal from this red motor neuron, you're expecting this muscle fiber 1, 2, and 3 to all respond. If there's a signal from motor neuron number 2, you're expecting this, cell A and B to respond. Yeah? Great. The most basic, simplest contraction you could get a muscle to do, just to like beep, is called a muscle twitch. That's saying you've had one action potential for a motor neuron. One action potential motor neuron comes, hits the muscle. What does the muscle do? It's called muscle twitch. Okay. Muscle fiber is going to contract and then relax really pretty quickly. This is interesting to me. Muscle contracts faster than it relaxes, which you might know. Also, if you've ever had a Charlie horse, that's not a muscle twitch, but you kind of, somebody ever had a Charlie horse? Right, your muscle just like cramps down, you're like, oh, okay, right? Yeah. Contraction and muscle twitch is faster than being able to relax it back out. As a muscle twitch happens, there are 3 parts to it. The first little bit, there's no muscle tension seen. So, like, signals are being sent down to the muscle, right? Boom, electricity, all the things, but there's no tension here yet, right? Like, it's working on it, but it hasn't got there yet. You don't start to actually increase tension in muscle until you get those cross bridges going, right? Acetylcholine moving or having the action potential move down muscle fiber, really important, but that's not actually causing contraction yet. Those cross bridges, that's where you start to, feel that force. Tension increases, and so long as you're still, contracting, great. Once calcium gets pulled back into the SR, right, you're going to relax the muscle. So it's going to look something like this. A myogram is just a fancy way of saying visual representation here of this twitch. We've got tension up here in no units, but just a little bit, a lot. And then this is time. Okay? This is, yeah. So, we got the stimulus. It contracted. The tension rose really, really quickly. And then over time, it relaxes back. One more thing. Even though the action potential is either like on or off, right? Muscles behave differently. They've got different properties. They've got different enzymes. They have all sorts of different things. This probably doesn't surprise you, but like a muscle contraction in your eye, or right there, eye, is here this blue line. The duration can be a lot shorter. It's rapid and short, right? Calf muscles are going to they're going to contract more slowly and they hold longer. The timing's just slightly different. I mean, these are all really fast, but like, your eye can twitch faster than other things. Concerns about that? Alright, that's what we've got for today. You have time for your quiz, 50 minutes long. Oh, I do want to ask you something while I'm taking attendance here. And this goes for online people too, so feedback is good. How was the timing on the exam? It was okay? Okay. I realize there's like, some people like to take a fair bit of time. I have one thought, and let me know if it's weird. I was wondering about, for the next exam, making it 2 exams, making it one, the multiple choice short answer part that's timed really, really tight, right? Just like, and then having another one that's the essay portions that's got a little bit more leeway. Is that annoying? Is that helpful? Does anybody care? All in one test? Okay.
Yeah, Sean last year for our physics final, he split up the exam into like 3 parts.
Yeah.
And it was timed, and it was kind of a mess.
It was a mess? Okay.
Yeah, because then you're worried about time, and then you got to switch. I don't know.
Okay. Freak people out?
Yeah.
Okay. It's one of those things that's like, it seems like the obvious solution, but then maybe not because, yeah, okay. But people were okay. I do realize it's pushing it on time. Or not. Oh man, my freshman class, I love them. I do love them. The person who got it was timed an hour, the person who got the worst score took like 18 minutes, and I was like, I didn't do so well. Yeah, because you probably didn't read the questions. Like, yeah. All right, that's it. Thanks. This is the kind of feedback I'm looking for.